Cellular respiration is a set of reactions that help make energy for cells. There's anaerobic and aerobic cellular respiration. The equation for this is C6H12O6 plus O2 turns into 6CO2 plus 6H2O plus ATP. There are 3 steps involved: glycolysis, the Krebs cycle, and the electron transport chain. The first step of cellular respiration, glycolysis, is when one glucose, 6-carbon molecule, breaks into 2 pyruvates, 3 carbons. The 2 carbofragments of pyruvate turn into acetyl-CoA. The Krebs cycle, the second step, is a series of 8 reactions in the mitochondria that takes in acetyl-CoA. Right before the Krebs cycle begins, pyruvate turns into acetyl-CoA. In this reaction, pyruvate, a 3-carbon molecule, is decarboxylated, which means a carboxyl group is removed, and it forms a 2-carbon acetate. The carboxyl group is released as carbon dioxide. This reaction is catalyzed by the enzyme pyruvate dehydrogenase. The enzyme pyruvate dehydrogenase then catalyzes the reaction, adds a CoA enzyme, and turns it into acetyl-CoA molecule. In the first step of the Krebs cycle, 2 carbon acetates that were formed from the pyruvate in the preceding reaction are attached to a 4-carbon molecule called oxaloacetate. This reaction is a condensation reaction. The joining of the acetate to the oxaloacetate requires energy that comes from the removal of the coenzyme A from the acetate. In the next step of the Krebs cycle, citrate is rearranged to the molecule isocitrate. In the next reaction, isocitrate is oxidized to alpha-ketoglutarate. As it is an oxidation reaction, an electron is added to NAD to release NADH. A molecule of CO2 is also released. This officially forms alpha-ketoglutarate. In this step, the 5-carbon alpha-ketoglutarate is converted into the 4-carbon succinate. A molecule of CO2 is also released. The remaining 4-carbon molecule is then converted into oxaloacetate, the molecule needed to condense with acetyl-CoA in Krebs cycle 2. The excess energy is conserved by adding coenzyme A to the succinate, forming succinyl-CoA. This energy will be harnessed in the next reaction when coenzyme A is removed. In this step, coenzyme A is removed from the forming succinate. ADP is then phosphorylated into ATP. Next, the succinate is oxidized to fumarate. Since it's an oxidation reaction, energy is released. However, there isn't enough energy to reduce NAD to NADH, and so another electron acceptor is used. FAD accepts electrons and becomes FADH. In this step, the molecule fumarate is rearranged into malate. This step is considered the last reaction because the next reaction requires the input of another molecule of acetyl-CoA so that the cycle can continue once again. In this reaction, malate is oxidized into oxaloacetate, the 4-carbon molecule that we use to join with acetyl-CoA to form citrate. Since the electron acceptor for this reaction is NAD, another NADH is formed during this reaction. Since we just made another molecule of oxaloacetate, we begin another cycle in the addition of acetyl-CoA.